everyone. This is GATE 2023 Life Sciences, Section P Biochemistry, question number 29. When cell components are fractionated by sedimentation, the correct order from lower to higher gravitational force G in which the components get separated is dash. A. Nuclei, mitochondria, microsomes and ribosomes. B. Microsomes, mitochondria, ribosomes and nuclei. Option C. Nuclei, ribosomes, mitochondria and microsomes. And option D. Ribosomes, microsomes, mitochondria and nuclei. This is the NPTEL online course reference to understand the theory behind this question. So cells could be fractionated in order to isolate each cellular organelle based upon their sedimentation rates. So cell has different internal organelles like nucleus, mitochondria, microsomes, and for our experiments, if we wanted to isolate mitochondria or nucleus from the cell, this technique fractionation by sedimentation is used, and we can sediment different cellular organelles by various centrifugation techniques like differential centrifugation and density gradient centrifugation. So the rate of sedimentation of each organelle depends on the properties of the each organelle, and the internal cellular organelles have different properties like shape. The shapes of the organelles are different, like nucleus. A nucleus is circular, mitochondria is oval, ribosomes are of different shapes, etc. Also, there are different size. The size of the organelles are also different. The molecular weight of the organelles is also different, and density of each organelles is different, which help us to fractionate each intracellular organelles depending upon these properties. For example, let's consider nucleus and mitochondria. So nucleus is very tightly packed with DNA and various proteins. In mitochondria, we also have mitochondrial DNA, but the amount of nuclear DNA is very large as compared to mitochondria. Hence, we can say nuclei is much more denser as compared to that of mitochondria. So, as I mentioned earlier, that we could fractionate the cells and sediment them using various centrifugation technique. So this is a tissue or an organ, and this tissue and organ is first homogenized. In this process, the tissue is crushed finely into a paste, and then this paste is further homogenized to disrupt the plasma membrane, and this process is known as homogenization. And then this homogenate is collected into a test tube, which will be further subjected to various centrifugation techniques. So this homogenate contains different organelles, and when we homogenate this tissue, the outer cell membrane of the cell is broken, and the organelle membrane is intact. So there are various different centrifugation steps. Different centrifugation techniques could be used. We could use either differential centrifugation, wherein the speed of the centrifugation, where the force, that is the RPM, rotations per minute, or the G. So in differential centrifugation, in the differential centrifugation, the speed that is RPM, rotations per minute, or G, rotation per minutes, or G can be changed. For example, there are centrifugations where you can keep rotations per minute to be 1000, 10,000. Similarly, we can also set the G value to be 1000, 15,000, 20,000, etc. And this technique of centrifugation is known as differential centrifugation. Okay. So as I mentioned earlier, that nucleus is very dense because it is very tightly packed with DNA as well as proteins in the DNA. So the centrifugal force required to, this is the centrifugal force required to sediment nucleus will directly be proportional to its mass. Okay, so when we remove the proportional sign, we'll have mass and G. G is a constant here. So as you can see, mass corroborates to the density of the organelles. So mass and G are inversely proportional. So if mass is higher, the value of G will be lower. Okay, this makes sense, that if mass is higher or density is higher, the G will be lower. So in simple sense, if we have if the molecular weight or the density is higher of nucleus, it will require less G to the bottom of the tube. So what I mean by this is denser or higher the molecular weight of any organelle, it will require less G for it to get sedimented to the bottom of the test tube. Okay, so nucleus is denser with larger molecular weight, so it will settle down at lower G, followed by mitochondria, followed by microsomes, followed by ribosomes. Ribosomes are the smallest. They have as compared to nucleus, ribosomes are smaller in shape, as well as since they are smaller in shape, their molecular weight is also low, their density is also very low, hence they will require higher G. They will require approximately 1,50,000 G for it to settle down. So while doing the experiment, what we do is this. So first this homogenate is taken inside the test tube, and it is centrifuged at 1000 G. So at 1000 G, we have nucleus which will settle down here. Okay, then the supernatant is collected into another tube. This whole supernatant is collected into another tube. And when we use 20,000 G, we'll have mitochondria getting collected. Again, the supernatant will have microsomes and ribosomes here, which will be collected in another tube, test tube. And when we use 80,000 G, we'll have microsomes getting sedimented. The supernatant then again will be collected, and finally we'll use 1,50,000 G, and we'll have very small tiny ribosomes getting sedimented. So this technique is known as differential centrifugation. So coming back to the question, they have asked us the correct order from lower to higher gravitational force. So as I mentioned earlier, nucleus is denser. So the formula was, this is centrifugal force. Okay, this is centrifugal force. Okay, and this is the G. So nucleus is very dense. It will require less G. It will require only 1000 G. Followed by mitochondria. It will require 20,000 G. Followed by microsome. It will require 80,000 G. Followed by ribosomes, which are very tiny, and they will require 1,50,000 G. So the answer is A, nucleus, mitochondria, microsomes and ribosomes. So they have asked from lower to higher gravitational force. So lowest will be nucleus and the highest will be ribosomes. So the answer is A. Thank you.